Good morning, everyone. Today the Journal Club is on the topic of the Klebsiella pneumoniae, expressing the WIM1 metallo-beta-lactamase is resensitized to cefotaxime via thiol-mediated zinc chelation. It is presented by the Dr. Maitri, and presenter is myself. This article is published in American Society for Microbiology in Infection and Immunity. This article is by the Harappa Karadutor and so nowadays there is a rapid increase of the antimicrobial resistance is of high concern with the ispic pathogens in which there is Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. Hospital-acquired infections by these pathogens are problematic, where the development of antibiotic resistance limits the treatment option. Development of the new antimicrobials and the combination treatments and modification of the contemporary antibiotics need to be addressed. So, over the past 2 decades, a new hypervirulent. Our main article is on the Klebsiella pneumoniae. So we need to be focused on Klebsiella pneumoniae only. So over the past 2 decades, a new hypervirulent Klebsiella pneumoniae has emerged as a clinically significant pathogen. Approximately half of all the hypervirulent Klebsiella pneumoniae infections occur in the young and healthy individuals and in young and immunocompromised individuals also. The hypervirulent Klebsiella pneumoniae strain is mucoviscous, typically because of the overproduction of its polysaccharide capsule and the presence of the virulent gene like RMPA and MAGA gene, which is most important in the K1 capsular serotype. The large virulence and plasmid LVPK encodes for the aerobactin siderophores. Expression of the RMPA is regulated by the availability of the iron. The hypervirulent Klebsiella pneumoniae can cause serious community-acquired infections in the healthy individuals. The hypervirulent Klebsiella pneumoniae was reported initially in Taiwan and Southeast Asia in the mid of 1980 and in 1990s. The hypervirulent Klebsiella pneumoniae is now the most common cause of the pyogenic liver abscess in Asia, and it shows intrinsic resistance to ampicillin. Dissemination of the mobile genetic elements conferring the antibiotic resistance and the antibiotic resistant hypervirulent Klebsiella pneumoniae isolates are increasingly being reported nowadays. Emergence of these strains poses a great threat to the public health also. If we see the epidemiology of the hypervirulent Klebsiella pneumoniae, it is found, the first case was found in Taiwan, China, Southeast Asia, Singapore. The rate of development of new drugs is low, while the resistance mechanisms are evolving rapidly. In particular, gram-negative bacteria like Klebsiella pneumoniae, they show the extensive drug resistance against many clinically used antibiotics and thus constitute the significant clinical challenges. One of the most important resistance mechanisms utilized by the Klebsiella pneumoniae is the expression of a specific beta-lactamase enzyme that hydrolyze and thereby inactivate the beta-lactam antibiotics. The beta-lactamase can be categorized according to the Ambler system with class A, C, and D, and comprising the serine beta-lactamase and class B comprising metallo-beta-lactamase. So for the today's topic, we are just focusing on the metallo-beta-lactamase. So according to the Umber-Bush-Jacobi classification, in the Umber group, they classified it as a class A, B, and C, and D. So we mainly focus on the class B, which is metallo-beta-lactamase. And according to Bush and Jacobi, the metallo-beta-lactamase is considering in the 3, in which the most beta-lactam includes the carbapenems, and it is representative enzyme for this is the IMP1, WIM1, NDM1, CCRA, and BCLL, B1, CPHA, B2, and L1, B3. And all this is inhibited by the clavinate except the class 3. So, according to this class B is sensitive with the penicillins, first generation cephalosporins, and second generation cephalosporins and third generation cephalosporins. But in our article, we generally focused on the second generation cephalosporins, and it is inhibited by the EDTA, which is a zinc chelator. So the zinc dependence of the metallo-beta-lactamase has been studied with the zinc chelators such as EDTA or depicolonic acid, which are able to resensitize the bacteria to beta-lactam antibiotics. In the therapeutic setting, they chelate not only the zinc but also a broad set of divalent metals, which can lead to toxic effects on humans. So that's why we just able to see it in vitro cultures, but not in vivo. Zinc chelation is an interesting approach against the MBL-mediated antibiotic resistance, and they have shown efficacy in vitro and in a mouse model, but not yet been developed or evaluated for the human use. So, given the potential benefit of the zinc chelation as a novel treatment option against the MBL-producing bacteria, it is relevant to search for the endogenous molecules with this capacity. This study was set out to search for an endogenous inhibitor of the metallo-beta-lactamase by screening the cell culture supernatants for factors that could restore the susceptibility of the MBL-producing Klebsiella pneumoniae to cefotaxime. So now I invite Dr. Metri for the article proper.
Good morning. Good morning, teachers and friends. The journal club topic assigned today is Klebsiella pneumoniae expressing WIM1 metallo-beta-lactamase is resensitized to cefotaxime via thiol-mediated zinc chelation. This was published in Infection and Immunity in 2019. The work has been done by Karadotti et al. Introducing this topic, there's a search for inhibitors of metallo-beta-lactamase, and the authors did this by screening cell culture supernatants for factors that could restore susceptibility of metallo-beta-lactamase producing Klebsiella pneumoniae to cefotaxime. Metallo-beta-lactamases require zinc ions in the enzyme's binding pocket to catalyze the hydrolysis of the amide bond in the beta-lactam ring, resulting in inactivation of the antibiotic. So zinc chelation is a possible strategy to resensitize Klebsiella pneumoniae to beta-lactams. EDTA and dipicolonic acid resensitize bacteria to beta-lactams via zinc chelation in vitro and in mouse models, but effects on humans is toxic. Therefore, the need to search for endogenous inhibitors of metallo-beta-lactamase. So to search for endogenous inhibitors of beta-lactamases, Klebsiella pneumoniae producing various metallo-beta-lactamases such as KPC, WIM, and also OXA48 and NDM was cultured in RPMI medium plus 5% urea broth with or without cefotaxime. And also Klebsiella pneumoniae producing these beta-lactamases was cultured in supernatant from HT29 colonic epithelial cells, which had earlier been cultured in RPMI with 5% urea broth with and without cefotaxime. The growth was monitored via bioscreen instrument and verified by viable CFU counts. Then to see if resensitization was specific for cefotaxime, the WIM1 producing Klebsiella pneumoniae strain A015200 was screened for susceptibility to ciprofloxacin, azithromycin, phosphomycin, imipenem, amoxicillin, doxycycline, cefotaxime, and ceftriaxone in RPMI plus 5% urea broth and supernatant with 5% LV. Coming to the procedure for bioscreen growth measurement, in this, colonies from overnight culture on blood agar were picked and cultured to exponential phase, that is, the optical density measured at 600 nanometers was 0.3 to 0.5 in LB at a pH of 7.5 at 37 degrees on shaking incubator. The bacterial suspension was then centrifuged at 5000 RPM for 3 minutes, washed phosphate buffered saline and diluted to an optical density of 0.05. For each well, total starting inoculum was fixed to 5 microliter from an OD of 0.05 in a final volume of 150 microliter of RPMI medium, which includes the various treatments which the authors have done through the course of this study in a honeycomb plate. The bioscreen instrument registered the optical density at 600 nanometer measurements every 10 minutes for 24 hours at 37 degrees centigrade and such that a growth curve was produced from each well. This was further verified by viable CFU counts. In this bacteria, that is 1 into 10 to the power 5 colony-forming units were suspended in RPMI or supernatant in final volume of 150 microliter in combination with various treatments in 96 well round bottom plates. The sample was incubated at 37 degrees on a shaking incubator for 4 hours. Then serial 10-fold dilutions of each sample were made and every dilution was plated onto the same blood agar plate, each plate being divided into 8 parts. The plates were incubated overnight at 37 degrees and colonies were counted. So we see that this is the result of the first part of the study. This shows that all bacterial strains were resistant to cefotaxime in RPMI medium with 5% LB. However, 2 strains producing WIM1 metallo-beta-lactamases displayed high susceptibility to cefotaxime when incubated in the supernatant of HT29 colonic epithelial cells with 5% LB. In these graphs, we can see that the purple line represents supernatant with 16 milligram per liter of cefotaxime, and whereas the blue line represents RPMI, that is in 5% LB, and the green is supernatant with 5% LB, and the red one is RPMI plus 16 milligram per liter of cefotaxime. Further, this was verified by the CFU survival assay, in which WIM1 producing Klebsiella pneumoniae strain A015200 was cultured in supernatant of HT29 cells with 5% LB and cefotaxime. So here we can see T0 is the initial inoculum, but we find that when this bacteria is grown in the supernatant along with the presence of cefotaxime, it is showing a 4-log reduction in CFU counts compared to culturing in RPMI medium plus 5% LB and cefotaxime. Then to see if this resensitization was specific to cefotaxime or that it took place with other antibiotics as well, we find that it was seen only in 3rd generation cephalosporins, that is cefotaxime and ceftriaxone showed regained activity against bacteria cultured in the supernatant of HT29 cells plus 5% LB compared to the RPMI medium with 5% LB. So coming to the discussion for the first part of the study, a secreted component in the supernatant of human colonic epithelial cells, HT29, resensitized WIM1 producing Klebsiella pneumoniae A015200 to cephalosporins, suggesting direct involvement of beta-lactamases, and the New Delhi metallo-beta-lactamase expressing Klebsiella pneumoniae did not exhibit restored susceptibility. It is possible that the NDM expressing Klebsiella pneumoniae had the presence of extended spectrum beta-lactamase plasmid, which was compensating for the inhibition of NDM by the supernatant of the HT29 cells. Then coming to the next part of the study, the authors wish to determine if zinc chelation could resensitize WIM1 producing Klebsiella pneumoniae. As we well know that zinc is a cofactor for this enzyme, so if zinc chelation could resensitize WIM1 producing Klebsiella pneumoniae strain A015200, so in this WIM1 producing Klebsiella pneumoniae strain was grown in RPMI plus 5% LB with and without EDTA, with and without cefotaxime, and the CFU counts were measured. Further, the Klebsiella pneumoniae strain grown in RPMI with 5% LB and in the supernatant, that is of HT29 cells with 5% LB, was serially supplemented with divalent metals, that is ferrous sulfate, zinc sulfate, magnesium sulfate, and calcium chloride at the concentration of 0.5 millimolar to see the effect that these divalent metals may have on the resensitization of the Klebsiella pneumoniae strain, and the CFU counts were measured. Following this, we know that EDTA can cause a breach in the can cause a change in cell viability by affecting the cell permeability. So this effect had to be seen if this effect was taking place. So for this, cytox green cell permeability was measured for EDTA and compared to heat-killed bacteria. Then free labile zinc was measured in the RPMI plus 5% Lb and in supernatant of HT29 cells with 5% Lb. Along with this, adding of zinc sulfate at various concentrations, that is 1.25 micromolar, 2.5 micromolar, and 5 micromolar. And EDTA was used as a metal chelator, and this was the positive control. So coming to the cytox green assay, cytox green is a high affinity nucleic acid stain. As the stain only penetrates cells with compromised plasma membrane, it is used to confirm loss of bacterial viability. So cytox green viability assay to assess if EDTA had any effect on the permeability and viability of Klebsiella pneumoniae. So Klebsiella pneumoniae strain A015200 was cultured in LB to exponential phase and diluted to an optical density at 600 nanometers of 0.1. Samples with 10 microliter of bacteria and 500 microliter broth with or without 0.1 millimolar EDTA were incubated for 1 hour. Positive control sample prepared by heat killing the bacteria at 70 degrees for 10 minutes. Then the samples were centrifuged at 10,000 G for 2 minutes, washed in PBS and resuspended in 2 molar cytox green in phosphate buffered saline. So after 5-minute incubation in dark, fluorescence was measured at an excitation wavelength of 488 nanometers and an emission wavelength of 523 nanometers. Coming to the results of this experiment, coming to figure A, we find that susceptibility of WIM1 producing Klebsiella pneumoniae A015200 was restored when cultured in RPMI medium plus 5% Lb with EDTA and cefotaxime at 16 milligram per liter. Then the susceptibility of WIM1 producing Klebsiella pneumoniae to cefotaxime was lost when cultured in supernatant with 0.05 millimolar zinc sulfate. As we can see in figure B, the supplementation with other divalent metals did not have such a drastic effect as the supplementation with zinc sulfate. Further, the EDTA did not have any direct effect on permeability of the cell as shown by the cytox green assay, in which we can see that the relative fluorescence of the strain itself and strain with EDTA are comparable, whereas the heat-killed Klebsiella pneumoniae showed a greater relative fluorescence. Then to detect reduced amount of free zinc in the HT29 supernatant compared to RPMI medium when fixed concentrations of zinc sulfate were used. So we see that when 1.25 micromolar of zinc sulfate is added to RPMI and the same amount is added to the supernatant, there is a definite decrease in the free zinc in the supernatant, and this trend is seen at higher concentrations of added zinc sulfate as well. So the discussion for this part of the study is that the restored susceptibility of WIM1 producing Klebsiella pneumoniae to cefotaxime was zinc dependent. Therefore, an active zinc chelator in the cell supernatant was causing the resensitization of WIM1 expressing Klebsiella pneumoniae to cefotaxime. Then the authors wish to identify factors in cell supernatant that could contribute to observed resensitization. For this, the HT29 supernatant was filtered through a 3 kilodalton size exclusion column. The filtrate was further passed through a reverse phase column and calibrated in 0.1% trifluoroacetic acid to enrich for peptides and proteins, and the first 15 fractions were obtained. The column was eluted with 0.1% trifluoroacetic acid in 80% acetonitrile. Then WIM1 producing Klebsiella pneumoniae strain A015200 was cultured in RPMI supernatant. The less than 3 kilodalton and greater than 3 kilodalton fractions of the supernatant, the less than 3 kilodalton flow through from the reverse phase column, and acetonitrile plus trifluoroacetic acid elute from the reverse phase column, and all these were grown in the presence of cefotaxime and also without. So in this part of the experiment, we see that the supernatant fraction, which was less than 3 kilodalton, retained the resensitizing activity to cefotaxime, as we can see over here. Then the less than 3 kilodalton supernatant flow through from the reverse phase column also retained the resensitizing activity to cefotaxime compared to the trifluoroacetic acid and acetonitrile elute from the reverse phase column. This shows that the sensitizing factor, which was chelating zinc, was strongly hydrophilic. Coming to the RPMI medium, this is a growth medium used in cell culture, and the name is derived from the Roswell Park Memorial Institute. Traditionally, it is used for growth of human lymphocytes. The authors analyzed the composition of RPMI to look for possible molecules which could be chelating zinc. So the composition of RPMI, 1 liter contains glucose and pH indicator, phenol red, and salts. And among the amino acid, they found this amino acid, that is cysteine. Furthermore, we know that the cysteine has intact disulfide bridges, which get reduced to L-cysteine during cell culture growth. And these cell cysteine residues have free thiol groups, and these help in the zinc binding properties of proteins, as shown by the various papers. So to study the effect of L-cysteine on WIM1 producing Klebsiella pneumoniae and its effect on susceptibility to cefotaxime, the WIM1 producing Klebsiella pneumoniae A015200 strain was cultured in presence of L-cysteine and L-cysteine at the concentration of 0.5 millimolar with or without 16 milligram per liter of cefotaxime. The viability was determined by colony count assay, and the growth of Klebsiella pneumoniae A015200 was measured in bioscreen C with cefotaxime and at varying concentrations of L-cysteine to determine lowest concentration for resensitization. So in this, we find that addition of L-cysteine to RPMI medium with 5% LB restored susceptibility of WIM1 producing Klebsiella pneumoniae to cefotaxime, whereas addition of L-cysteine to RPMI medium did not restore susceptibility of WIM1 producing Klebsiella pneumoniae to cefotaxime. This part of the experiment shows that the lowest concentration of L-cysteine that affected antibiotic resistance was 125 micromolar. As you can see, that 62.5 micromolar of L-cysteine did not have any effect on the growth curve, whereas as soon as 125 micromolar of L-cysteine was taken, it affected the antibiotic resistance. So reduction of L-cysteine to L-cysteine in supernatant is important for inhibition of WIM1 activity, and addition of L-cysteine studied in bioscreen system indicates that the lowest concentration of L-cysteine affecting antibiotic resistance is 125 micromolar. Then, to further validate the role of L-cysteine in WIM1 Klebsiella pneumoniae susceptibility to cefotaxime, the authors used Delbecque's modified Eagle medium complete or DMEM without these following: L-cysteine, methionine, and glutamine, and these were added to the HT29 cells. Then the supernatants from these, along with L-cysteine in sterile water, were passed through reverse phase column, equilibrated in 0.1% trifluoroacetic acid, and the fractions were obtained. The thiol content of processed supernatants and L-cysteine in sterile water was examined in each fraction using a GSH standard. The Klebsiella pneumoniae strain cultured in fractions 6 and 7 of DMEM and DMEM incomplete and L-cysteine in sterile water with or without cefotaxime, and then colony count assay was performed. The thiol measurement assay was done by means of a fluorometric thiol assay kit. Fractions from reverse phase column were analyzed, and the thiol content determined was based on glutathione standard curve. Fluorescence was measured with an excitation filter of 490 nanometers and an emission filter of 520. Coming to the result of this part of the experiment, we see that DMEM complete showed thiol content in fractions 6 and 7. Pure L-cysteine in sterile water also showed thiol content in fractions 6 and 7, whereas DMEM without the presence of cysteine, methionine, etc. showed absence of thiol content. The colony count assay of Klebsiella pneumoniae strain A015200 cultured in presence of fraction 6 and 7 with CTX showed resensitization to CTX only in fraction 6 and 7 of DMEM complete and L-cysteine in sterile water, whereas the fraction 6 and 7 of DMEM without cysteine and methionine did not have the resensitizing activity. So fraction 6 and 7 from DMEM complete, supernatant, and pure L-cysteine in sterile water resensitized Klebsiella pneumoniae to cefotaxime, and this corresponds to the thiol content of these fractions. No resensitization was seen in fraction 6 and 7 from DMEM, which was deficient in cysteine, methionine, etc. Therefore, presence of free thiols regulates susceptibility of WIM1 producing Klebsiella pneumoniae to cefotaxime. Further, the authors wish to see that if any alternative thiols had a similar resensitizing activity on Klebsiella pneumoniae to cefotaxime, so they used glutathione. So Klebsiella pneumoniae strain cultured in RPMI with glutathione, which was added either in oxidized or reduced forms, and colony count assay was performed. The supernatant was collected from HT29 cells and analyzed for reducing property using the conversion of fluorescently labeled L-cysteine to L-cysteine. And then they wish to see whether this PX12, which is a molecule which specifically inhibits thioredoxin pathway, had any effect in the conversion of cysteine to cysteine in the supernatant, which was measured by the fluorescently labeled L-cysteine conversion to L-cysteine conversion to L-cysteine, and relative change in fluorescence was noted. So in this part of the experiment, we find that oxidized glutathione had no activity on bacterial resistance to cefotaxime, whereas the reduced form of glutathione had the same activity as L-cysteine, as we can see in this graph, that L-cysteine and glutathione reduced in the presence of cefotaxime show a definite decrease in the colony count. Then, to confirm cell-dependent reduction of L-cysteine, the authors took only RPMI, cells plus RPMI, and then cells which had been cultured in RPMI for 24 hours, so the cells plus supernatant and supernatant. So they find that there is an increase in fluorescence in cells plus supernatant as well as the supernatant, which means that there was a cell-dependent reduction of L-cysteine to L-cysteine. Furthermore, inhibition of the thioredoxin pathway with PX12 added in varying concentrations to the supernatant showed a decrease in fluorescence as the concentration of PX12 increased. So thiols such as reduced glutathione have the same activity as L-cysteine in resensitizing Klebsiella pneumoniae to cefotaxime. Reduction of L-cysteine to L-cysteine occurred in a cell-dependent manner, and the thioredoxin pathway is involved in generating free thiols in the cell culture supernatant. As we can see, the thioredoxin reductase it reduces the thioredoxin, and this in turn reduces the protein which has a disulfide bond and causes the formation of a free thiol group. So now the authors wanted to see expression of New Delhi metallo-beta-lactamase 1 in a neutral background to confirm resensitization of cefotaxime using L-cysteine. So in this case, NDM1 producing E. coli DH5 alpha isolate was tested for cefotaxime susceptibility in the presence and absence of L-cysteine, and this was again measured with bioscreen growth curve. So here we find that there was a 1,000-fold reduction in the MIC of DH5 alpha isolate after 10 hours, which is comparable to the study on Klebsiella pneumoniae. Then, to study the change in the susceptibility of WIM1 in a neutral background, the authors conducted a transconjugation experiment. This was a two-step strain transconjugation was performed, transferring the WIM1 plasmid from A015200 to E. coli ATCC strain 25922 and an MG1655 wild-type strain through a CAM-R Dept intermediate strain. The transfer was confirmed by colony PCR and gel electrophoresis. The Vim enzyme was successfully transferred to the recipient strains, as we can see here. So the growth of the Vim1 transconjugates was again measured with a bioscreen assay in the presence of L-cysteine and cefotaxime, and we find that the L-cysteine is again resensitizing it to cefotaxime. So the result of this experiment is that the final recipient strains tested in bioscreen instrument for susceptibility to cefotaxime with or without L-cysteine showed that Vim1 activity was affected by free thiol in L-cysteine, and Vim1 activity was not related to the genetic background of the bacteria. Then, to ascertain if zinc chelating thiols in urine determines cefotaxime susceptibility of Vim1 producing Klebsiella pneumoniae, urine samples were collected from healthy volunteers centrifuged at 4,500 rpm for 10 minutes at 4 degrees, and samples were subjected to size exclusion and reverse phase fractionation. Colony count assay with Klebsiella pneumoniae strain in RPMI medium plus 5% LB plus minus cefotaxime plus minus urine was performed. The thiol content of fractions was measured with fluorometric thiol assay and compared to GSH standard. Then the urine samples were further diluted from 100% to 5% in PBS, and its effect on bacterial growth was seen in the presence of 16 mg per liter of cefotaxime. Pure or diluted urine, diluted to the concentration we just showed resensitization, was used to culture Klebsiella pneumoniae strain in presence of cefotaxime with or without zinc sulfate, again to see the effect of zinc sulfate. So we find here that in the first figure, on incubation in urine, susceptibility of Vim1 producing Klebsiella pneumoniae was restored. Then fractions 6 and 7 of the urine were able to resensitize bacteria to cefotaxime, and this corresponds to the earlier results in the earlier part of the experiment. Here we can see that fractions 6 and 7 of the urine samples were able to resensitize Klebsiella pneumoniae to cefotaxime. And fractions 6 and 7 again of urine contained thiol, suggesting presence of L-cysteine or other thiol containing molecules in the urine. Then urine concentration required to sensitize Klebsiella pneumoniae to cefotaxime for each donor sample was found to range from 20 to 50% of urine, and the urine sample, both 100% as well as diluted to a concentration that still showed resensitization to cefotaxime, was supplemented with zinc sulfate, and then the effect was noted. So we find that when zinc sulfate is added, it prevents the resensitization of the Vim1 Klebsiella pneumoniae strain to cefotaxime. So in this study, bacteria regained susceptibility to cephalosporins only, suggesting thiol-mediated zinc chelation and metallo-beta-lactamase inactivation. Effect on imipenem by zinc chelation initially was not observed, which suggests a more complex mechanism in metallo-beta-lactamase resistance against carbapenems. Also, the New Delhi metallo-beta-lactamase expressing clinical isolates did not exhibit restored susceptibility, possibly because of the presence of an ESBL plasmid, which compensated for inhibition of NDM by EDTA. Free thiols were not originally present in cell culture, but were formed by extracellular reduction of L-cysteine to L-cysteine via the thioredoxin pathway. This reduction via thioredoxin pathway exposed the free thiol group in cysteine, which is crucial for the interaction between cysteine and zinc, allowing the chelation of zinc. To rule out that the effect was related to the genetic background of bacteria, Vim1 plasmid was transferred via transconjugation to one ATCC strain and to one MG1655 wild-type strain, where resensitization to cefotaxime was seen in the presence of L-cysteine. This supports the fact that the effect was directly related to the Vim1 plasmid. And this study is of great significance because it points to the fact that urine contains free thiols, which can be regulated by various factors, including inflammation, drugs, and diet, and which in turn can be useful in resensitizing bacteria to antibiotics. The urine itself did not have the ability to reduce cysteine to cysteine. Use of EDTA is not a viable option for treating antibiotic-resistant bacterial infections due to its toxic side effects. So host factors such as L-cysteine and free thiols may be a key solution against resistance in combination with antibiotics. Thank you.
One question is, what is honeycomb plate and what are its uses? Bantuni?